the class on drug development. You guys have probably had this class several times before, but it's a very, very important portion of participating in the pharmaceutical industry. So having a discussion about drug development is sort of important. My name is Darshan Kulkarni. I'm the principal attorney at the Kulkarni Law Firm. In ways of introduction, I have a PharmD, I have a JD, and a Master's in Quality Assurance and Regulatory Affairs, and I'll be teaching the class. Let's sort of talk about drug development. What are we going to talk about? We're going to talk about the goal of this presentation. What is the goal of drug development? Then look at the divisions: preclinical, IND, phase 1, phase 2, phase 3, and then talk finally about the NDA, or sometimes as may be referred to as the BLA. What is the goal of this presentation? The goal of this presentation is to first outline this presentation, like we just said, then identify issues that may be of concern, and then discuss them. We will do more of the discussions in class, but more of the outlining and explaining during these recorded lectures. What will we not be covering? We will not be covering history. That's really boring. The laws. Yeah, I can talk about the laws, but no one really wants to hear about them. International drug development, we could talk about it. It's primarily the same thing, not quite sometimes. We aren't going to talk about combination products. We aren't going to talk about the different FDA meetings that happen. And we won't talk about the generic drug development pathways, which may be actually of interest to some people. Conditions, let's sort of recognize this. Every drug must meet 4 criteria. It must be safe, it must be effective, or also known as efficacious, and those are slightly different terms. They mean slightly different things. Effective just means in general it should work, but efficacious means it should work for the indication that we're talking about. And every drug must not be adulterated or misbranded. Let's recognize I do have a typo in there. What are the divisions of the FDA? It's CDER, CBER, and CDRH. CDER is for drugs, CBER is for biologics, and CDRH is for medical devices. Before we go into that, we should recognize, because this question comes up quite often, what is the difference between CDER and CBER? CDER refers to small molecules. Small molecules are things that are like aspirin, small, tiny, usually less than 15 to 20,000 Daltons. On the other hand, CBER is for large molecules. Molecules like hemoglobin, molecules like thymoglobulin, that would typically be produced by biological systems. So they tend to be anywhere from 50,000 to hundreds of thousands to millions of Daltons in size. And then there's CDRH, which is the medical device area. What is the drug development process in a nutshell? What you're looking at is the drug development process. It starts from synthesis and purification. It does animal testing. You get the involvement of institutional review boards. These animal testing can be short-term or long-term. And then you actually enter into phase 1, phase 2, phase 3. We could talk about accelerated development, treatment INDs, or parallel track, but that's not going to be the focus of this talk. There are different FDA meetings that are scheduled depending on when you do it. You might have advisory board meetings. You might have opportunities to answer questions. But that's where you are for the most part. Screening, you're using a couple of different chemical techniques, things like combination chemistry, high throughput screening. This is usually done to identify, okay, we've got 10,000 potential candidates. How do we narrow it down to 5 or 10 that we're going to pursue? That doesn't mean that all of those 10 have a good chance of succeeding. In fact, I think it's out of every 10,000 potential candidates, only 1 actually gets all the way through. These are just different ways you'd identify them. Then there's preclinical, so you'd actually do tissues. You'd test the potential molecule on tissues, then you test it potentially on organs. You'd then test it on small animals and large animals. The idea is you want to avoid testing in living systems until such point as you know that it works, to whatever extent you can know that it works. The idea is you give the drug a little bit to the tissue, see how the tissue reacts, and you give it a little bit to the organ. It's a more complicated system. How does the organ actually respond to that? Then you talk about animals, small and large. A small animal wouldn't be similar to a human being, but you get ideas of how a complicated system sort of responds to it. And then large animals like beagles and chimpanzees and larger animals. and how does a larger animal respond to it? This is obviously a very, very ethically problematic area. Is it ethically appropriate to be testing on animals? Some would say no, but the question is, if you don't test on animals, are you okay with human beings being the first living organism to have something tested? A lot of people like to say, well, we can test in computers. Computers are that smart. No, they aren't. People like to pretend that they are. They really haven't gotten to that point. They aren't even close, because we don't understand the systems well enough yet. What is the IND filing? So now you've tested this potential product in animals, and you're going to go to the FDA and now say, I would like to start testing this in human beings. You provide an introductory statement, a general investigation plan saying, here's what I intend to do in human beings. You provide an investigator's brochure. You talk about the protocols that you want to use in these human beings. You talk about, okay, here's how we've been making this thing. Here's what we know about this potential product. So that's the chemistry, manufacturing, and control information, or the drug substance characterization information. You can say, here's how we think that this drug potentially works. Here are the potential toxicological experiences. In certain situations, you may actually be in a situation where this drug was previously used. It may be in other countries. It may be for other indications. And you might talk about the previous human experience. What is the process in a typical clinical trial? Your pharmaceutical company is usually the sponsor. That sponsor will go out, talk to someone like a CRO, or a clinical or contract research organization. These guys are the ones who manage the study for you. That doesn't mean that the sponsor is not responsible. It just means that the CRO has specialized expertise that they're being pulled in for. The sponsor remains responsible for the study, unless there are some delegation of responsibilities. That CRO will say, I need to talk to these different sites. I want them to actually do the study for me. These are doctor's offices or hospitals or something like that. Sometimes it may be easier to actually just directly work with what's referred to as an SMO, or a site management organization. A site management organization is represents some, many, all doctor's offices or sites, and they're supposed to theoretically simplify and streamline the process of drug approvals and doing clinical trials. Unfortunately, the experience has not been positive, so SMOs have been falling out of favor. There has been a slight resurgence recently, but for the most part, they aren't the most popular thing in the world. You then do a phase 1. A phase 1 is where you actually outline what you're going to do. The idea is this is your first in human beings. You have very, very small numbers, anywhere from 10 to 20 patients. Actually, I shouldn't call them patients. We should actually call them subjects. A patient is one who actually has a legitimate expectation that they may get better. When you're doing clinical trials, you don't actually have a legitimate expectation that you will get better, because we don't even know what we're giving you. A well-done study would actually be randomized, double-blinded, placebo-controlled, multi-center, multinational studies. And we can talk about what each of those are. The goal in a phase 1 is really just to say, here is our first experience with human beings. Are we going to hurt someone in a very significant way? This is obviously a very, very scary thing to do. So who are people involved in phase 1s? They're usually men. They are usually younger men. Why men? Because, quite honestly, in many cases, you don't have to deal with fluctuating hormones in the case of women. So statistically, you have fewer variables. Like we said, we're talking about a small number of patients. Phase 2, the goal for phase 2 study is really to evaluate primarily safety. So you know you aren't hurting anyone. You know you aren't killing anyone or maiming someone. What you don't know is what happens if you give it to larger populations, and do we know enough about it? So you'd give it to maybe anywhere from 100, 200, 300 patients, and your goal is to see what are the typical adverse events you would observe. In a phase 3 study, on the other hand, your goal is to give it to 3,000, 10,000. I've seen studies up to 60,000 patients. And the goal of those studies is to show efficacy. So far you've just been trying to make sure you don't hurt someone. Phase 3 is really making sure that this drug actually does what it's supposed to do. So the goal for phase 3 study is testing efficacy. the numbers are substantially higher, the statistical requirements are substantially higher, but that tells you if the drug works. At that point, you go to the FDA, you talk about wanting to sell the drug in the US. You must show that it's safe and effective. You must show that the risks are outweighed by the benefits. You must show that the labeling is appropriate. You must identify methods and controls to show strength, purity, drug identity, quality. And that's the end of this presentation. If anyone has questions, feel free to email me. I look forward to hearing from you. Take care. Bye-bye.